So our first speaker is Dr. Gus Tonelli. He's an assistant professor in computer science and engineering. And I'm going to have him to maybe introduce himself and the topic of his presentation today. Forrest, it's all yours.
Great. So can you all see my slides?
Yes.
Great. So my name is Forrest Tonelli, and I'm an assistant professor at the University of South Carolina, Department of Computer Science and Engineering, as well as a faculty member at the AI Institute. And today I'm going to be talking about explainable artificial intelligence in the Rubik's Cube. And I really wish I could stay the whole time, but I'm actually running the CSC seminar, and that's coming right after this, so I gotta leave. But I wish I could stay to see everybody's presentation. So I'm going to give some motivation, and I know this is the genomics breakout, and some people might be wondering, well, why are you talking about the Rubik's Cube? And so I want to give some motivation, not just why we should study the Rubik's Cube for AI, but what this means for all other different types of fields, and how the Rubik's Cube is actually deeply connected to these different types of fields, including fields in the natural sciences. Then I'm going to talk about the algorithm DeepCube, how we were able to use it to solve the Rubik's Cube, and how it's lacking in the ability to actually explain to us its strategy for solving this. And then how we want to move to an explainable artificial intelligence method, particularly one that is collaborative, where a human is in the loop and can collaborate with this explainable AI system to find solutions, personalized solutions that they themselves understand. So the Rubik's Cube itself is a pathfinding problem. And so when I say pathfinding problem, I'm saying that there's a problem in which we are at some configuration, or as we say, some state of the world, and we want to find some sequence of actions that can transform one state into another state. So for the Rubik's Cube, we have some scrambled cube, and we want to find a path to the goal where all the faces have the same color. But this is actually many problems in robotics, natural sciences, and mathematics can be posed as pathfinding problems. For example, in theorem proving, I can start from some premises and through some sequences of actions, namely some inference rules, I want to find a path to some logical statement that I want to prove. In chemical synthesis, I may want to find a path from some building block to a molecule that I want to synthesize. For program synthesis, I may want to find a path in the space of programs that is going to write a program that accomplishes some task. And then in robotics, I may want to find a path to manipulate an object, or to assemble an object, or to move around a room. So these pathfinding problems are seen, throughout robotics, mathematics, and the natural science. So the reason we focus on the Rubik's Cube is, one, there wasn't any domain-independent AI methods that were able to reliably solve the Rubik's Cube up until this point. And so this was already a challenge for AI, but the reason it was a challenge is because there are many different configurations, over 10 to the 19 possible configurations. So you can't just brute force a solution. You have to actually find a solution in a intelligent manner. And we are doing this without any domain-specific knowledge about the cube. So we are trying to have some domain-independent method that can actually solve these pathfinding problems. So not only do we use the Rubik's Cube, but we use other puzzles as well. And then we hope to transfer this over to different domains, such as problems in theorem proving and chemical synthesis. And then furthermore, there's been a lot of work on solving the Rubik's Cube in AI using planning methods and pattern databases, and using knowledge about group theory, so on and so forth. So people have put a lot of effort into this task, and there are ways to actually find what's called a shortest path, so solving the cube in the fewest number of steps, that exploits a lot of domain-dependent knowledge about the Rubik's Cube. And so we can use this to evaluate our domain-independent method. So in the work with DeepCube, we investigated 7 different types of puzzles and 4 different categories. So one is a Rubik's Cube. The other is a sliding tile puzzle. And so there were just 4 of these puzzles of different sizes. And the one with the largest state space is the 48 puzzle. So that has 10 to the 62 possible combinations. And then also this lights out puzzle where if you push a square, that square will be toggled from on to off, as well as the squares immediately horizontal and vertically adjacent to it as well. And the goal is to turn all these lights out, as well as this soccer band puzzle. So you're this little green agent, and your goal is to push these boxes onto these red targets. And the core method that we use is value iteration. So what we really want to learn is how to solve this problem in the most efficient way possible. So if you're thinking about something like chemical synthesis, you would want to synthesize a chemical that is going to be, for example, maybe the least expensive way, the way that consumes the least amount of resources possible. So when we're talking about pathfinding problems, we're often interested in a what people call shortest path, lowest cost path, or an optimal path. So here's an example I show my students in my AI classes. And imagine you're this green tractor here, and you want to get to this shovel, and you want to do it without running over any rocks or any plants. Say you're on some AI farm here. So of course you don't want to run over rocks, it'll damage the machine, and you don't want to run over plants because it'll damage the crops. And so you want to find a path that is going to reach the shovel without wasting too much time. And what value iteration is able to do is it is able to actually initialize some table. For example, say there are 100 states here. This is a 10 by 10 grid. And in this table, we have some initial estimate of this cost to go. And what's the cost of a shortest path? We have some initial estimate. And then we can update this estimate using the Bellman optimality equation as an update rule. And essentially what this is doing is I look at every action that I can take, and then I'm saying, what's the cost of taking that action, and what's my estimated cost to go from the resulting state? And I use this to then update my estimate of the cost to go. And I do this iteratively until the cost to go for all of the states stops changing. And then in that case, I can be guaranteed to converge to the true cost to go. So I will have found what's the cost of a shortest path for every single state in my state space. And then I can find an optimal path just by behaving greedily with respect to this cost to go function. So at every state, I look at every action I can take and say, what is the state that I can transition to that has the lowest cost possible? And I transition to that state, and I continue to do this until I've solved the problem. So here's a visualization of this. In this case, this is showing the negative cost to go, but it's equivalently the same thing. And you can see here's the goal and how this update process propagates outward from the goal. Let's just run it. You see with each step of value iteration, it's able to find shortest path for more and more of these states. Then I'll just fast forward a bit more. And then here it has converged to the true cost to go, so the true cost of shortest path for every state in the state space. So if we want to find, solve problems in the most efficient way possible, solve these pathfinding problems, whatever it is, they're improving chemical synthesis, all we have to do is use this value iteration method. Now the issue is when, this is a small problem and it has 100 states, but when we get to situations where we're dealing with very large state spaces, we just don't have the time or the memory to store everything in the table and go through every single possible state and update that state. So for example, these puzzles, chemical synthesis, problems in quantum computing, when you formulate these as pathfinding tasks, the state space is so enormous that you just can't possibly do this value iteration on any modern day computer. And so then what we're going to need is some type of approximation. So we're no longer going to store every state in the state space, but we want some approximation architecture that can map the state to some estimate of its cost to go. And we hope that this approximation architecture has vastly fewer parameters than there are states in the state space. So the approximation architecture that we use is neural networks. And there are a bunch of different approximation architectures out there. There are decision trees, support vector machines, so on and so forth. It turns out on certain types of problems, neural networks perform very well. And indeed, with the Rubik's Cube, this was one of those problems where the neural network was a very good approximation architecture. So neural networks, just a brief overview, are inspired, have inspiration from the brain. Now, these aren't exactly what, this isn't exactly what happens in the brain, they're just inspired by the way the brain works. And they are composed of neurons that have incoming connections and outgoing connections, and those connections are known as weights. And then a neuron can fire based on its activation. And this activation is dependent on the signals that are propagated forward through the incoming connections. And when it fires, it sends a signal to other neurons via its outgoing connection. And so neural networks saw a lot of early success when it came to recognizing handwritten digits and these types of things. But when it came to more complex tasks, for example, natural image recognition, so trying to distinguish between, say, for example, different animals in the wild just from natural pictures and things like that, neural networks oftentimes fell short. But given better hardware and more data, this has become doable with neural networks, but particularly by modifying the architecture such that we stack these layers of neurons one on top of the other. And these form what is called deep neural networks. And so these are very good at learning from very low level features, for example, like pixels or something like that. And so one of the reasons that people believe deep neural networks perform very well is because they're able to form this hierarchical structure of the data. So here is a paper that is showing neural networks learning something about, for example, faces or cars. And you'll see in these different layers, they learn more higher and higher level features. So in the first layer, they'll learn very low level features like edges. In the second layer, they'll learn higher level features like eyes, what's an eye, what's a nose, so on and so forth. And then in the last layer, they'll learn about entire faces. And so this is thought to be the reason for the success of these neural networks. And so deep neural networks have found a lot of success in all sorts of different areas. So one of the earliest is in this image net classification task here. And so that set off a lot of excitement about deep neural networks. And we can see that they've been used in the natural sciences, from high energy physics to chemistry to bioinformatics. Furthermore, they've also been used in tasks that are involving a sequence of decisions having to be made, for example, playing video games, playing board games, solving puzzles, and robotic manipulation. So the architecture we used for this project is essentially the same across all puzzles. The only difference is the input here. So the input is the representation of each puzzle. And for example, for the Rubik's Cube, there are 54 stickers on the Rubik's Cube. Each one can be 1 to 6 colors. And so we just use a one-hot representation. So just 54 times 6 gives us a representation of 324. And we essentially use this one-hot representation for all of the puzzles. So the only thing that really had to be changed in this algorithm is just describing the input to the neural network and describing how the puzzle moves. And then after you have described that to the system is able to solve these problems. So like we saw before with value iteration, we saw that the learning an optimal path was propagating outwards from the goal. So it started from the goal, and the learning essentially propagated outwards. Now, we don't have enough memory to store every possible state, but we can sample states from the state space to train our neural network. And the question is, which states do we sample? And so we take inspiration from the way value iteration works and start from the goal and randomly generate states. So we can randomly generate states by scrambling the goal 0 to n times. And then this can ensure that if we scramble it fewer times, that we have states that are closer to the goal. And if we scramble it more times, we have states that are further and further away. So through our approximation architecture, it allows us to propagate this signal from the goal to states that are further and further away from the goal. All right, so approximate value iteration is doing the same thing as value iteration, except instead of using a table, we're actually using this to train an approximation architecture. So we'll do one iteration of this update, and then we'll use our neural network to summarize this update. Then our neural network takes the place of this J, our estimate of the cost to go, and we will repeat this process again and again. And so then we can get an estimate of the cost to go. Like we saw before in value iteration, we can also try to solve the problem by behaving greedily with respect to this cost to go function. So we can look at every action we can take and we can move to the state that has the lowest cost. Now, when we do this with the Rubik's Cube, we have our neural network and we're behaving greedily with respect to this neural network, which represents our approximation of the cost to go. We see that we don't reliably solve the problem. So in this graph, we have on the x-axis, the training iteration, and on the y-axis, we have the percent of cubes that are solved. And each line represents a different number of times the Rubik's Cube is scrambled. And so you see, after about scrambling at about 7 times, it starts to do worse and worse at actually solving this cube. So if you scramble it 10 times, it only reliably solves it about 90-some% of the time. You see as we scramble it 15 times, it only solves it about maybe 45% of the time. And then if you scramble it more times, like 20 and 30, it's even less than that. So we see that it's learned something, but it's not able to reliably solve this problem. So we need something in addition to this to allow our agent to solve this problem. And so we can then try to integrate this cost to go function with search. And the reason search can help us is because search allows an agent to think before it acts. So it thinks about what would happen if it took some action before it actually takes that action. So before, when we were just behaving greedily with respect to this cost to go function, it was just taking actions without thinking. It just used the same rule every time to take an action. But now we can actually anticipate the results of the actions that it can take, and then based on that search, act in the real world. And so what we can do is we can use this cost to go function as a heuristic that can tell us how close we are to solving the problem. And so this heuristic function is used in what's called A-star search. So A-star search is a very popular algorithm in the pathfinding, in solving pathfinding problems. And also, if the heuristic meets certain criteria, there are some theoretical guarantees. Now, because we're dealing with this complicated nonlinear function in a neural network, we can't have these guarantees on the shortest path, but we'll see in the results that we often still find the shortest path despite not having these theoretical guarantees. So the way A-star search works is that it prioritizes, it does this search in the state space, and it prioritizes states that have a lower cost over states that have a higher cost. And cost is computed by summing up two features of a state. One is the path cost. The second is the heuristic. So the path cost is the cost from a path from that starting state to some state X. And then the heuristic is the estimate of the cost to go. So the cost to go from X to the goal. And so if you sum this together, this is an approximation of the total cost of that node. And since we want to find paths that have the lowest cost possible, we're going to prioritize nodes with a lower cost. And so we're going to repeat this process of selecting a node and expanding it, looking at all of its children, and then evaluating the cost and putting them into this priority queue until a node associated with the goal state is selected for expansion.
Forrest, can you hear me?
Yes.
Yeah, sorry, my audio was having issues, so I had to switch computers. But do you have your PowerPoint presentation in presentation mode? Or does it matter, or are you going to manually change it? I can see it just fine.
Oh, is this not, are you seeing on the side as well?
The slide that I'm seeing is motivation applications. I think it's possibly one of the very first ones, slide number 8. Is that the correct one?
No. Can you see this integration with A-star search?
No.
Okay.
It might. be that somehow screen is frozen if you want to just refresh it. We do have time. We started the session a little bit early, so don't rush through it. Just take your time and see if you can debug this.
Okay, can you see this now?
Integration with, base search.
Okay. Oh, my apologies. Oh wow, I was. So I just wanted everybody to just see this video. Hopefully, this will clear up what I was saying about value iteration, but how this propagates from the goal outwards. So as you can see here, this is propagating from the goal outwards in terms of how to behave. All right. So essentially, this is what we're doing to train our neural network. And so I guess some people missed all of this, but this is a neural network. And our training data starts from the goal and propagates outwards, so on and so forth. Okay, so I was just finishing talking about A-star search. And so with A-star search, we are just prioritizing nodes by the cost. So we have our path cost and our heuristic, which comes from the neural network, which is trained with approximate value iteration. And so A-star search typically expands one node at a time, but because we have this deep neural network, which can be computationally expensive, we can expand multiple nodes at a time and use GPUs to provide us this parallelism. And so by combining this method of this approximate value iteration in A-star search, we can then try to solve these puzzles, and we can also compare these puzzles to the cost of an optimal path. This isn't computationally possible for all the puzzles, but for the ones that are, we compare it to the cost of an optimal path. We can see that it solved all the puzzles in question. And then when it came to finding an optimal path for the Rubik's Cube, it was able to do this 60% of the time. For the 15 puzzle, 99% of the time. The 24 puzzle, 96% of the time. And for the lights out puzzle, 100% of the time. So even though A-star search has these theoretical guarantees, if the heuristic function meets some admissibility criteria, which we can't guarantee for this neural network, we still see that it was able to find an optimal path a lot of the time. So this is very promising for when we're using real world, an algorithm such as this for real world applications, and we want to save costs, for example, when we are doing chemical synthesis or we are doing robotics and we don't want to waste resources. We want to find a path that is an optimal path or close to an optimal path. All right. So here is a demonstration of our work. I think this should be running this server right now, but this is a demonstration of the Rubik's Cube being scrambled and then solved. And so you see it's able to find a solution. You can tweak the search parameters such that it can find a solution really quickly, even though the solutions are a bit longer. And so it finds a solution in about a second. And when this work came out, there was some attention in the media and things about it. And I was very excited about this optimal path, but instead what happened is people were so excited about seeing that it was able to solve it in 1 second. And I didn't really cross my mind that it was interesting, but a bunch of other people found it interesting. Okay. And so people have also used this algorithm for other applications, for example, applications in quantum computing as well as cryptography. So I don't know too much about what's going on in these fields, especially the quantum computing, but apparently you can pose some problems in these domains as pathfinding problems and then just apply this algorithm to these problems as well. And so that was really exciting to see. Okay, so even though that we can use this algorithm to solve the Rubik's Cube, we can't actually use this algorithm to teach ourselves how to solve the Rubik's Cube. Say, for example, if I didn't know how to solve the Rubik's Cube, I wouldn't be able to actually learn how to solve the Rubik's Cube by just watching this algorithm. It doesn't give me an overall method for solving it. So the part of the reason this is the case is because we use these deep neural networks, which are these complicated nonlinear functions. So it's really hard to interpret what's going on. And also we use the search methods, for example, in this case, A-star search, which are just doing this, looking ahead and seeing, planning different paths and finally finding a goal, but it doesn't have any type of explainable element to it. So if we do some digging, for example, in the Rubik's Cube domain, we can see that this knows something about symmetry. So for example, if you give it mirrored states, it'll often find mirrored solutions to mirrored states. So here are two mirrored states, and you see that the solution it finds is actually a mirror solution. So maybe it knows something, or in the neural network somewhere, there's something about symmetry. It also knows something about these conjugate triplets, which are come up a lot in group theory. For example, you do some action A, some other action B, and then reverse that action A prime. So this is called like AB A prime. And it does these conjugate triplets often. And so we had to actually know this beforehand and do some digging, but how could it actually tell us about this, even if we didn't know? Similarly, when people use deep neural networks and search to play games at superhuman levels, for example, with AlphaGo, it's able to perform really well. Of course, it's playing at the superhuman level, but it's not able to explain its strategy. So for example, here is what's called move 37. And this piece here was moved by the agent, but nobody was able to really understand this move. And actually people thought that it was a mistake or that it was a bad move, but it actually ended up winning this game, and it was playing one of the best Go players in the world. And so since it ended up winning this game, this is probably a good move, but people were just confused about why it would do such a move like this in the first place. But it's not able to explain anything to us. It's just using some neural networks to guide a search procedure. That's essentially what it's doing. And in these applications, in these different methods, DeepCube-A and AlphaGo, it doesn't actually know if you ask it, for example, how many stickers are on the Rubik's Cube. I can't ask it that. I can't ask it which pieces are in the correct position. Similarly with Go, I can't ask it, even if you ask it something simple, we could all do, count how many pieces there are. It won't be able to just tell you. So there's a lot of basic things that it is not knowing. So it's able to solve these problems, but it doesn't know basic things about the problem. So we want to use explainable artificial intelligence, not just to be able to solve these problems, but to explain them in a way that humans can understand. And so we are taking inspiration from the way the Rubik's Cube is typically explained to humans. So you have this sequence of sub-goals, and then you have these algorithms to help you achieve each sub-goal. And we want to not just find some fixed set of sub-goals and algorithms, but to be able to allow a human to interact with the system and find a particular solution that they themselves are interested in. Or maybe the AI algorithm wouldn't be able to solve the problem without some human intuition guiding it. And then we can create this positive feedback loop where humans are learning from AI and AI is learning from humans. And so here's a visualization of the architecture we're working on for the Rubik's Cube. So a human can interact with the system through a chatbot or a visual interface, and they can pose their own ideas or ask questions about how to solve the Rubik's Cube. And then using some explainable algorithms on top of these unexplainable ones, in particular knowledge graphs and logic programming, we can then generate solutions that a human can actually understand. So we can hopefully generate this natural language and show concrete examples to explain to a human how to solve the problem. And so, like I said, even the basics we'll have to explain to the system. So we have to explain it what the different pieces are, what the centerpiece, edge piece, corner piece, what a layer is, so on and so forth. And from these very basic concepts that we take for granted, hopefully it'll be able to come up with an explanation. And so, for example, if we just want to solve this very simple case of getting this one edge piece in place, if we do a naive approach, for example, using the decision tree, which are often thought of as interpretable in machine learning, you'll see that this decision tree is very large, even though it's just asking two questions. Which side is the white sticker facing, which side is the orange sticker facing, and then it tells you what to do based on that. but this isn't very useful. But something more intuitive, like get at least one pair of stickers to match. If the white stickers are matching, then turn the white face into the orange stickers do too. Or if the orange stickers are matching, turn the orange face into the white stickers do too. This is something that we can understand that's very short and concise. And so this is our goal, to generate these short, concise statements that we can actually express in English. And so we have a preliminary work on this at a AAAI demonstration. So we have a demonstration of this interface where it actually is going to explain how these algorithms for solving, in particular, the white cross of the Rubik's Cube. And then I just want to acknowledge my students that are part of this project, as well as my collaborators. Okay, here's some references, and that's it. Thank you.
Thank you, Forrest. We're going to take a few minutes to see if there are any questions in the audience. So if you do, please unmute your microphone and go ahead and present your question. If not, I actually have a couple things I'd like to ask, but I'll give the audience first shot. Maybe I can ask my question. So first, I can imagine there's quite a bit of parallel between, pathfinding, the optimal pathfinding, and problem solving in the areas of, for example, drug design, as well as, so for example, I can see how path interaction, protein genomic path interaction as well. Can you comment what would be needed from your perspective? What data would you need in order to apply this technology to the domain of health?
Okay, so when it comes to, for example, drug design, so to define something as a pathfinding problem, one of the most important parts is defining this transition function. So I'm in some state, I take some action, what happens next? And I think I have a visualization of this. In chemistry, this can be decided partially with rules of chemistry, but sometimes it's a bit more difficult than that. And so deep neural networks are actually used for chemical reaction prediction. So, what happens if I have some reactant? What would the product actually be? And then if we can actually define this, then we can build off of this to actually train a neural network to have some cost to go function, and then use A-star search to solve the problem. But actually defining what this transition function is can be really difficult.
I see. So I can imagine another application of this would be, so not in order to transition people from a state of, let's say, disease to the state of health, it's not a single step transition, it's multiple step transition, which at every step, multiple decisions need to be made. What kind of food you need to eat, what kind of exercise you need to do, what kind of sleep you need to get, what kind of stress handling mechanisms you're gonna. So do you imagine this application, that would be another application of this technology to essentially would be a lifestyle coach sort of a thing?
It's possible. But the more I think about these problems, the more I think we have to deal with the uncertainty of these problems. So with, for example, the Rubik's Cube, we know it's a deterministic domain. So we know exactly what's going to happen. But in these other domains, many possible things can happen. And they can happen with different probabilities, and they can have different values for their outcomes. So some could be a lot better, some could be a lot worse. And so you have to take all of this into account when you are actually trying to find some sequence of decisions that you're going to take to get a person to be healthy. And so, I think there's a lot of, I think it's far away from actually being able to simulate exactly what would happen if, someone does some exercise regimen or eats a certain way, to actually be able to apply this to that scenario, as well as the fact that this isn't just a sequential decision-making problem. Maybe you could pose it that way, but you're taking multiple decisions at the same time. You could be exercising and eating healthy at the, choose that at the same time. So yeah, I just think it's a long ways away. What I'm really interested right now is just getting chemical synthesis to work, and then worrying about these other problems a bit later. Yeah.
I see. All right, well, thank you very much. Unless there are any more questions, I know Dr. Agostinelli is under time constraint. He has to be at a different seminar in a few minutes. So if there are questions, maybe a quick question. Otherwise, Forrest, thank you very much for your presentation, and hope to see you soon again.
Yeah, thank you. Sorry about the slides, I didn't notice.
Oh, no problem. I should have intervened sooner, but my computer was having audio issues on this end. So by the time I closed one to get to the other one, it just kind of took a little bit of my time too. So, okay. Well, thank you very much.
Thank you.
See you, talk to you later. Bye.
Bye-bye.
All right, I'm gonna move on to our next speaker. So our next speaker is Dr. Mohamed Azar, who is an associate professor of cell biology and anatomy in our School of Medicine here at the University of South Carolina. I'll let him introduce himself and the title of his work. So I'm excited to hear what you have to say, especially if it is going to go anywhere in that inexplicable circadian rhythm. I'm actually very, very much interested to hear more details. So thank you very much. Mohamed, it's all yours. Again, 25 minutes. I will let you know 10 minutes and 5 minutes ahead of time. I will send it by chat. I'll let you know those time stamps.
All right. Can you see my screen? Okay.
Yes.
All right. Good afternoon, everyone. I'm Mohamed Azar, and thank you for invitation and introduction, Dr. Belafar. So my lab works on mouse genetics and cardiovascular disease. And basically we are using, RNA-seq and microarray technology to look for differentially regulated genes during the development and progression of the disease, as well as we are now doing some experiment where we are able to reverse aortopathy and trying to find out if we can actually reverse the gene expression patterns, by treating these diseases. So today I would like to share some of the work that my graduate student is doing, basically to look for some of the genes that we have found in aortopathy. And the aortopathies, so this is, most of the work is done by Mengistu Gabri, an outstanding graduate student in my lab. So he's basically single-handedly did most of the work. So aortopathy is kind of a general term where we, we describe, for example, aortic aneurysm and dissection, hypertension, vascular calcification. So aortopathy comes in various forms. The project that I'm going to be talking mainly has to deal with the vascular calcification. And the vascular calcification is also of different kind. There are sometimes you will see, for example, atherosclerosis, which is a disease of arteries where you have these fat deposition. In those arteries, mostly you will see the innermost layer of the artery, which is called as intima, which is mainly where the endothelial cells are. You get plaque formation, and those plaques are calcified. But then you also have vascular diseases in patients such as diabetes, chronic kidney disease, hypertension. And in fact, even in old age, we get our vascular layers are actually calcified. And that kind of calcification you mainly see in the middle, or the most predominant layer, which is called as medial wall of the aorta, as well as the small peripheral arteries. So you will see essentially calcification or bone formation if you look at these patients. So for example, in this patient, you have this entire medial wall, but you'll see a ring of these bone as well as or calcified minerals that are being deposited in the medial wall. So what happens as a result of that, you will have, for example, if the resistance arteries where you get this calcification, it sort of leads to a lot of stress and eventually leads to hypertension. In other cases, for example, if you have calcification of an artery that is mainly peripheral arteries that supplies blood to the limbs. You can feel that it leads to, for example, ischemia, where the muscle is not able to get enough blood supply, and as a result, there will be problems healing wounds, etc. And in most cases, in fact, it's not very uncommon that you will see patients of diabetes and chronic kidney disease actually have to go through some limb amputation, which is kind of leads to a very morbid life. So unfortunately, there's no treatment. For most cases, surgical treatment is the only way, whether it requires extreme forms, as I said, you may have to go through amputation or some kind of endarterectomy to sort of allow the blood to kind of circulate well to the skeletal tissues. Now, as I said, the most pathological changes are restricted mainly to the medial wall, where you get the calcification, but there are a lot of systemic issues. You will see disbalance, for example, impaired calcium phosphate, if you look at the blood, and there are several systemic issues that you will see in these patients. So obviously, since there is no medical treatment, there's a lot of interest to use omics, proteomics, as well as genomics approaches to find out, the kinds of genes or targets that are involved in the pathology, as well as find out if we can, able to leverage that into some kind of therapy to target certain, key regulators of vascular calcification. So to folks who are not familiar with the vascular tissue, you have this is basically a diagram of an arterial wall. So you have the inside. So this is the lumen where the blood usually is. And this is a layer which is called as intima. It's made of endothelial cells. Then you have this medial wall where you have most of the smooth muscle cells are present. And that's where you have most of the elastic fibers are present, which allows for these blood vessels to contract and relax, which is the most important element in the vascular system. Because if you don't have that elasticity, you actually, if you calcify the tissue, eventually it leads to resistance, which also affects the cardiovascular. And most of these patients essentially not died because of aortic rupture, they actually died because of the stress on the heart. Ultimately, the heart has to work very hard and undergo failure. So if you have, for example, in this aortopathy condition, you will have a lot of this calcium deposition, mainly localized to the medial arterial layer in these patients. And what happens is you have a lot of elastin breakdown, and that elastin sort of attracts calcium, and the calcium kind of binds, and then you can see a lot of these smooth muscle cells then phenotypically change into what we call as osteoblast-like cells. So they become like a bone-like cells, which sort of create ectopic bone formation in the vascular wall. And that leads to other problems. For example, the intimal layer, which are the endothelial cells, also undergo dysfunction, which is very important for endothelial cells provide a lot of chemicals that are required for contraction and relaxation of the smooth muscle medial wall. So if you have endothelial dysfunction, that can contribute to the disease progression as well. And then you have adventitia, where you have a lot more collagen is produced in response to this disease in the medial wall. So you have a lot of adventitial fibroblasts, proliferation and thickening of the adventitia, and a lot of collagen. That further adds to the biomechanics issues. Basically, it makes the arteries very stiff, which eventually tend to rupture faster. So what we have done in the lab, basically, we use a genetic approach where we, for example, has some what we call as recombinase mice, and these are reporter mice, where we actually tag these mice in a way that only arteries, if we have a mice where we can only tag the smooth muscle cells of the vascular wall. So rest of the tissues in the body, for example, has the gene what we call as tomato. So all cells will be red, but only the smooth muscle cells that we are interested in and follow the fate of those cells will be green. So it will be easy for us to make any genetic changes or anything and follow the fate of those smooth muscle cells. So we use these mice, and then what we have, so this basically shows if you have a mice that you just put in a normal diet, and then you are actually mark the cells. So for example, this is the medial wall. You can see all cells that are green are smooth muscle cells. So the blue dots are basically nucleus of those cells. And the red cells are essentially cells that are non-smooth muscle cells. So this is the intimal wall. These are endothelial cells. And this is your adventitial fibroblast here, which are all in red. So basically, by looking at this, you can follow the fate of the smooth muscle cells. So what we have done, we had these mice and we subjected these mice to some adenine diet. So adenine diet is basically, it's sort of a model which induces kidney failure. So essentially, kidney is very important for calcium phosphate metabolism. As you can imagine, if kidney fails, there will be high amount of calcium phosphate circulating, which eventually leads to chronic kidney disease. So we actually are able to produce a mouse model for chronic kidney disease, which develops arterial calcification. So if you look at these mice that we fed like about 12 weeks of adenine diet, high phosphate adenine diet, you can have pretty much the whole arteries, the whole thoracic aorta, as well as abdominal aorta. And we have seen all these peripheral arteries. For example, you see these are the arteries that supplies blood to the leg. So you have all of these arteries and bigger or smaller arteries that eventually develop calcification. Now, this is a micro-CT. If you look at the micro-CT, you can actually look at the calcification in these light white color. So you'll see again, the aorta has calcification throughout the length of its aorta, thoracic and abdominal aorta. You will have some cases where you'll have, for example, abdominal aorta is more affected than thoracic aorta, for instance. So, but that's, for example, about 15% of the cases we see mild calcification. But most of the time, we are able to generate very severe medial arterial calcification by adenine diet treatment in 12-week treatment. The advantage to this model is now we have developed a protocol by which we can actually follow the disease progression in a live animal. So we can simply put the mice on diet and just keep repeating and follow mice by micro-CT analysis. So we can put the micro-CT and you can see at the beginning, this is a control mice. This is the mice that's being treated with adenine diet. So this 6-week treatment, as you can see, it starts to increase. As we go along the treatment, you can see the arteries start to calcify. So you can see nice abdominal calcification of these arteries here. So you can look at the other side. You also have renal arteries are calcified. A lot of mesenteric arteries are calcified. You have thoracic aorta is being calcified. So you can see that nicely. So we basically now have a model where we know when and where the calcification is. So we can actually find a way. If we can find a way to treat, we should be able to. The goal ultimately we are aiming is that there is no way you can treat once the arteries are calcified. So we're trying to use some approaches to decalcify the artery and see if we can actually follow them in our micro-CT, that first you develop the calcification and then we decalcify and you should go back to the normal. So that's really the entire goal of this project. So what we have done is we already know that we tagged the cells through this green color GFP in these mice. So if you have a controlled diet, you can see all nice smooth muscle cells in the medial wall. When we have the mice that are treated with the adenine diet, you have huge amount of calcification. So you can see there's a big calcification lesion here. And you can see that all cells where you have the calcification, most of these smooth muscle cells actually undergo apoptosis and they die. And that part is essentially replaced by this bone or the calcified material. So essentially, we identified the smooth muscle cells are really the major cell type that undergo, transformation and being replaced by calcification. So then what we did was we actually our collaborator in Clemson University, Dr. Narendra Bahari, he actually provided us some nanoparticles that has some EDTA loaded in these nanoparticles. And these nanoparticles are basically connected to an anti-elastin antibody. Now, the advantage is when we inject these nanoparticles, elastin antibody direct these nanoparticles to places where you have the elastic damage, which is really the places where we have the calcification. And it does not interact with native elastin. So therefore, if you have an artery that has a native and intact elastin, this antibody or nanoparticle will not go there. So essentially, by using that approach, we are able to target these nanoparticles. So we have developed an experiment where we have the control mice, which basically essentially mice on a normal diet that do not develop any calcification. Then we also injected these nanoparticles where we actually replace EDTA with a fluorescent dye R. which essentially will tell us where exactly the nanoparticle is. So we can do, for example, fluorescence imaging, and we can localize the presence of the nanoparticle to tell us if we are targeting it to the right places. We also added blank nanoparticle where we don't have any drug in it, and we just wanted to see if the nanoparticle by itself has any toxic effect. So we have those built in. And then in the main experiment, we have the EDTA nanoparticles that are injected to see if we can actually target the EDTA to the right places. Now, the EDTA we've done twice. So for example, if we have, we have the animal that first we tag with the smooth muscle with the GFP, then we basically put them on adenine diet, and in 12 weeks, these mice are calcified. And then we started injecting, after confirming with micro-CT that these guys do have calcification, we started injecting them. And we have given up to 16 injections of EDTA nanoparticle. And then we take the animals down and do the characterization and histology, and just to make sure, the calcification is gone. In the other experiment, what we have done is we calcified the arteries, we injected nanoparticles, and then basically we put them on a normal diet. By the way, these mice are on normal diet. So once we're done with the EDTA treatment, we switch mice to the normal diet to recover, because the diet itself has a lot of problems because it's pretty intense. So if you put them on normal diet, the mice actually are recovering, and you are injecting nanoparticle. In another control, what we have done is we injected nanoparticle, but then we actually stop the nanoparticle and allow the mice to go on to see if they actually get the calcification back. So we basically calcified the artery, decalcified them, and let them go to see if the calcification is going to come back after some time. So we have done those experiments as well. So basically, this is the first result of the first experiment where we loaded the nanoparticle with dye-R, fluorescent dye, which actually fluoresces in this infrared size 7 fluorescent range. So you can see the pink color here is the dye. So you have, this is the vascular wall, and this is the vasovisorum. So what we have found is that our nanoparticles are actually coming from the vasovisorum side. So they're not entering from the blood, although we did the intravenous tail vein injection. We're finding that these actually nanoparticles are finding its way to the artery through the vasovisorum. So you have a lot of these nanoparticles, as you can see, are being accumulated in the adventitial side of the vasovisorum of the vessel wall. So that just shows that, okay, I think we can load these nanoparticles, target them to the right side. And then basically what we did was we had these live images where these are the animals, live animals, and we did the micro-CT. For example, to the left is control. There's no calcification. There's no aorta that's calcified. But here you can see calcification in these aorta. So these are kidney that are calcified. And you can see there is aorta here that you will see that's highly calcified in this mice. And this mice, we started injecting nanoparticle, and this is basically after nanoparticle, you can see we're able to decalcify a lot of this aortic tissue that you will see here. So, and you can also appreciate that basically the calcification of the kidney is pretty much gone. So you'll also have, you can decalcify soft tissue, not only the aorta, you can also have some other soft tissue that might have been calcified can also, because you can see very strong calcification. You don't see that calcification in kidney here. So we are very excited that we probably are approaching a potential injectable therapy where we can actually avoid surgeries, and eventually that's really the goal that we want to do. So this is now the images, as you can see. So control, the thoracic aorta, you will see there's no calcification, so therefore you will not see any aorta in the micro-CT image. And you will see a serious calcification of the aorta. The entire aorta essentially was calcified in these mice. Now, when we injected, 16 injection can take off a lot of calcium from this aorta. So you can see now the aorta is quite perforated throughout its length. And when you measure the amount of calcium, you can see there's a significant drop in calcification of this entire aorta, as we see. Now we have done almost 6 or more animals that all shows a down modulation of calcium. Perhaps we can continue because we have just given 16 injection. That is good enough to show us significant reduction, but we stopped at 16 injection. In the other experiment where we allowed the mice after the injection, we stopped the injection and allowed the mice to go for several months. So we found that after 2 months, we have the calcium basically. So these are the mice that basically, these are calcified mice switched on the normal diet, and they never receive any EDTA injection. So basically there you'll see the calcification continue to increase. On the other hand, if we start an EDTA injection, we can actually suppress the calcification in these animals. So with that, what we did was next we said, okay, if that seems very promising, what about can we restore or regenerate the tissue? Because we know calcification has destroyed most of the medial wall. Can we regenerate and produce a new tissues that basically heal the damaged elastin? So what we have done here, so you can see this is the same control experiment. You will see a lot of calcification in the adenine dye treated mice. But if you have mice that is being treated with EDTA nanoparticle, you can see there are a lot of cells that are coming which are not smooth muscle cells. So now you will see that the tissue seems like regenerated quite a bit, but it's getting a lot of help from a lot of the adventitial side. There's a lot of cells are accumulated. There are a lot of these red cells that are essentially non-smooth muscle cells that are now finding its way and migrating and trying to heal the damaged tissue. So you can see there are a lot of these cells are here. You can look at these cells, and these cells they don't look like smooth muscle cells, because the smooth muscle cells is a very typical morphology. These cells look like more like a myofibroblast or some kind of cells which actually are more like a fibroblast. So they could have been macrophages, they could have been fibrocytes, they could have been some stem cells that are migrating in to sort of heal the tissue. On the other hand, we also found where you have an extreme amount of calcification, what mice has done is actually EDTA injection has split that calcium into smaller nodules. And by doing so, you will see there are a lot of cells that accumulate, for example, this nodule from the intimal side. And what we are thinking that there may be a lot of endo-EMT cells that are actually derived from the endothelial cells that are sort of attacking this nodule from the left. And from the right, from the vasa visorum, you have extreme amount of infiltration of these myeloid cells. So there are a lot of blood-derived cells are coming and trying to enter here in order to clean, and the area that are being decalcified can be actually filled with cells. On the other hand, we have also find there are cells which are actually smooth muscle derived because they are still green color, but they don't quite look like smooth muscle cells. They actually look like cartilage. So there are a lot of cartilage-like cells which are derived from smooth muscle cells are also being part of this healing complex. So you'll have a lot of these cells coming and trying to fill those gaps where the calcium once reside. So we're thinking that I think this EDTA nanoparticle not only decalcifying the tissue, but it's also activating the system, which is probably supplying cells to heal those parts. So it's really important to understand what's happening at the molecular level. For that, what we have done is we thought, okay, let's look at all the gene expression changes. So what happens when we get the calcification? What are the genes that are downregulated? What are the genes that are upregulated? So first we wanted to identify the gene expression program that is altered during the vascular calcification. And the next goal is basically that once we treated these with EDTA, then can we restore the normal molecular program? Can we revert those genes that we know are altered because of calcification? So that's really the overall goal and the purpose of doing this RNA-seq analysis. So with that, we have about 6 samples that we have, male and female control and experimental that we tried. And we have looked at about 10 to 15 million reads, which is probably more than sufficient to give us a good idea of what genes are going up and down in our experimental and control animals. So this just shows basically what we have done. We have a basic cutoff of 1.5-fold increase and decrease, and we picked up any statistics significance that is less than 0.05. So as you can see in this dot plot, you will have genes that we are focusing mostly is essentially the ones that we know are significant and statistically significant, and also have a fold change greater than 1.5. We find there are certain genes of interest that actually are also present, the genes that are statistically significant, but we are not paying attention to the genes that are not significant, or even if they have a fold change increase, we basically are not looking at them at this point because they're not statistically significant in every sample. So as you can see, for example, our top candidate here, which is upregulated, is called SPP1, which is essentially a master regulator of calcification, is called osteopontin. So that is already being shown in the literature in human patient that osteopontin is highly increased as patient who actually have chronic kidney disease, diabetes associated with vascular calcification. So is the TIM1 is associated with calcification, and there are some genes that we are learning are highly downregulated, have to do something, for example, with circadian rhythm. And that's what I think Homeo was trying to warn me. But we do have some genes that are basically suggesting that, yes, there may be some. And talking to our collaborator, Dr. Fong, just last week, we learned that there are patients, of, for example, peripheral arterial disease, who have altered sleep patterns. So it's possible that these mice, because of the extreme pain, etc., they're going through, that might be sufficient enough to change their circadian rhythm. So we did a lot of analysis, but I think for the time's sake, I'll just focus on the main crux of all the analysis that we have done. For example, one of the bases was we said, okay, we asked question, what are those genes that actually are altered or differentially regulated, are directly related to, for example, diseases of the arteries. So for example, arteriosclerosis, what are those genes that are involved in arteriosclerosis are also changing our mice? What are the genes that, for example, are seen in patients who have chronic kidney disease? Because we are actually causing chronic kidney disease to induce vascular calcification. And we found there are a lot of these genes that are very critical and actually are lighted up in our analysis, which confirm that I think we have some very key regulators that are involved in vascular calcification. And these genes basically, for example, you have smooth muscle actin here, which is a major actin in the smooth muscle wall. You have, for example, as I mentioned, there's MMP12. These are matrix remodeling enzymes, which are very important for remodeling the extracellular matrix in the vascular wall. We also ask a question, so the genes that we are having, what is their connection to overall cardiovascular health? So basically that goes back, as I said, the vascular calcification by itself is not really a bigger problem. In fact, what it does to the heart is a major problem because it just puts a lot of stress. It leads to cardiac dysfunction, heart failure eventually, hypertension, cardiac fibrosis. So you will see that a lot of genes that we are finding are actually connected to myocardial infarction, atherosclerosis. We have looked at genes which are arteriosclerosis. We have also found genes which is basically are seen in obstructive lung disease. For example, pulmonary hypertension is another problem that leads to heart failure. So you've seen a lot of those interesting genes that basically are again common with the chronic kidney disease. So these are some of the genes that are our primary targets, and we are actually looking at them more closely to see if we can actually learn something from them, and maybe those might become a potential target, in our new experiments. We have also done some closer look. For example, this is the kind of analysis that I was trying to do where we actually now have genes that we looked at different biological functions. So for example, what are the genes that basically are involved in extracellular matrix organization, genes that are involved in elastic fiber formation, genes that are important for extracellular proteoglycans. So these are some of the key elements that we see are changing in our mice. For example, levels of proteoglycans such as aggrecan is gone up. There are a lot of ground substances are increased in the vascular disease during calcification. We have seen elastic fiber formation is affected. So we are seeing a lot of key genes that are involved. For example, if you look at elastic fiber, you will find genes such as fibrillin-1, which is in fact the mutation in fibrillin-1 causes elastic fiber disruption and Marfan syndrome. So yeah, a lot of those genes that basically are affected, and tells us and provide us a lot of information about the biological processes that are being affected by this analysis. So there are two major centers that emerged that we thought are very interesting for the vascular calcification point of view. One is inflammation. As I said, there's tremendous amount of cells such as macrophages, myeloid-derived macrophages, and fibrocytes that actually migrate to the wall. And they also become source of a lot of stem cells. So there are a lot of genes that we know are altered in these and may be contributing to inflammation that we see. And of course, there are major genes that are involved in, for example, extracellular matrix organization. As I mentioned, fibrillin-1, LTBP2 is another gene that is major for elastic fiber formation. BMP4, for example, is very important for, it's being established that BMP4 plays a very important role in vascular calcification. There are studies who supports it. So you have thrombospondin, for example, that is also an important protein that is involved in activation of TGF-beta pathway, which is critical for vascular homeostasis. So we are basically identified a lot of those genes that we are now going after, in a very organized way, try to look at and validate them and see if they're somehow an important player. So we, for instance, so this is some of the key genes that I thought was important. If you look at the procalcitic genes, we have identified. So the genes in red are upregulated. The genes that are blue are downregulated. So we have seen, for example, osteopontin, osteonectin, osteoglycine. These are very critical genes that have known function in vascular calcification, are all upregulated. We have proteoglycans that we know are associated with cartilage. Remember, cartilage is the primary source that give rise to the osteoblast. So of course, osteochondrogenic gene expression is important. So we find agrican, which is a cartilage-specific proteoglycan, is upregulated. These are all cartilage-specific proteoglycans, and they're all upregulated. Lumican, for example. And there was a paper just came out that if you knock out lumican in mice, actually leads to aortic dissection and rupture. Elastic fiber assembly, as I mentioned, fibrillin-1 is an important gene that's downregulated. There are collagen fibers which are upregulated because, as I mentioned, one of the response to vascular calcification is the fibrosis. So you have a lot of collagen that actually being deposited in the adventitial side of the vascular wall. And we have clearly found that a lot of these genes are actually upregulated. There are enzymes that are very important for matrix remodeling, and they're also very important for osteoclasts. The osteoclasts are essentially the bone cells. These are basically giant hematopoietic stem cells that fuse and forms a bone resorpting cells. So these cells essentially remodel the bone. So if you have some kind of calcification laid down in the wall, osteoclast is constantly working and trying to remodel those calcific lesions. So we are seeing a lot of those genes that are involved in osteoclasts are highly activated in these mice. And of course, as I mentioned, there are genes that we are seeing a very systematic program that is being activated, basically depressed. So here, for example, NFIL3 aren't like one. These are aryl receptor, nuclear translocator, hydrocarbon receptor, nuclear transfer like gene. These are the transcription factors which are master regulator for the genes that are being regulated by in circadian clock. For example, PER1, PER2 being the target of these. So these are transcription factors that are downregulated, suggesting that some of the genes that are supposed to be suppressed by circadian clocks are basically activated in these mice. That confirms, perhaps, indicate that maybe this is another pathway to look how these changes in the circadian clock are feeding into the vascular calcification pathology. And we also identified genes that are directly involved and upregulated in TGF-beta pathway, which is also very important and is a known pathway for vascular or aortopathy. So what we are doing now, so this basically, I told you the baseline that we have already done RNA-seq on the animals that we know actually develop vascular calcification. So what we have done now is actually we injected these mice with EDTA nanoparticles. So and then after 16 injection, we collected these animals. So we have almost five animals that are ready that we collected that as we speak actually are being, we sent out these samples to a company who's actually doing RNA-seq on this. So once we get that data, the idea is basically we will compare those to the before the EDA treatment. So we want to know the genes that are differentially regulated by EDTA treatment. Can we actually reverse those gene expression back, and then can we restore the normal transcriptomic pattern of the of the control animal? That's really where we are heading, and hopefully we'll have this in a couple of months. So as I mentioned, that one of the things that we are very excited about, that I think we have an injectable therapy that seems promising in reducing vascular calcification in a mouse model of chronic kidney disease. And we have identified some key gene by simply RNA-seq data analysis. And we are now trying to do more RNA-seq analysis to see if we can actually reverse those gene expression that we have seen are being altered in these animals. And finally, and not the least, I want to thank Mengestu, who has done most of the work, and then my collaborator, Dr. Sue Lesner, who basically is spearheading the subcontract which funded most of these studies in collaboration with Dr. Naren Vaivahari. And we have a good team with Dr. Forrest and Home Belafar, and we also basically collaborating with Dr. Fong at Prisma Health to talk about these things. Essentially most of the work in mice, but I think we get a lot of clinical input and a lot of approaches where we can maybe start to think about, once we have these RNA-seq data, can we actually compare these to publicly available data, where we can maybe perhaps find more relevance to the clinical situation. And with that, I would like to thank you for your attention, and I'll be happy to answer any question if you have.
Thank you, Mohamed. That was great. That was, I have probably 10 questions to ask.
Sure.
But again, I'm going to wait to see if there is anything from the audience first. Thank you. So please feel free to ask your questions. Maybe I can ask one of the 10. I should have sorted this in the more important order, but so after the injection of the EDTA nanoparticles, you show that the calcification decreases
Yes.
in the media arterial aorta. So I'm curious to know, what is the effect of it in just regular calcification in the bony material? Is there any reduction of calcium in other places?
All right, this is something, in fact, I was just talking to my student that maybe we need to talk to you about this, because we have absolutely no clue how to look for that, whether we are, it's like we are also causing a collateral damage, right? So on the one hand, we are able to rescue the vascular calcification, but then if we don't want to, there are chelation studies that are being done where people have injected EDTA directly, and they found it leads to osteoporosis in the longer term. But I think we think there's a lesser of a chances, because our nanoparticles are basically gets out very quickly. They go to the kidney and the liver, and they're being cleared out. And most of them which is stuck is essentially the one that's targeted through these elastin antibodies. But nonetheless, we have to find a way to actually address that.
I see. So I have several other questions, actually. Do we know how long the nanoparticles actually stay in the system, or what is the mechanism of them being flushed?
This is, I think, mostly come from my collaborator. So Dr. Vevahari said that essentially it takes like a few days for these guys to clear. But we have done, for example, when we inject, and we looked at, I think, in 2 days, 1 or 2 days, 24 hours, we have done the study. And we took the entire animal and we put it in IVIS. And we are able to see, I think, that they were there in the liver. I remember clearly, even after 24 hours. But we haven't done longer term studies to see what happens to these nanoparticles. But I think Dr. Vevahari said that most likely they kind of cleared out in a few days.
I see. I see Dr. Puchivishek has turned on her camera. I presume she has a question to ask.
I do.
So I'll just pass it on to her.
Okay. Hi, Azar. It was a really interesting talk. So I'm really interested in circadian rhythmicity and sleep, and study this in animals. And I guess I was wondering if I understood correctly, all the samples were taken at the same time point, right? But yes, what you did find is these differences in circadian entrainment. So I guess maybe just a suggestion or a question for you of, have you considered to actually look at how that rhythmicity has changed, of just taking a few different time point snapshots and doing the analysis to see how much that rhythmicity is disrupted.
Do you want to write a grant together on this?
We can think about it, yeah, for sure. And I guess I do want to know is, were they actually taken at the exact same time, or was it during working hours? And I'll just fully admit that years ago, I never paid attention to this either, but now we certainly, pay very special attention to the exact time of day for, taking.
Yeah, so for the sample collection, most of our sample collections are done in afternoon. So that's really the time. We know that most of the time, by the time my student gets down to this, is I think around 3, 4 o'clock.
Right.
So that we sure. I don't know whether that has anything to do what we are seeing, and we are seeing consistently. And as I said, we never thought about this, and now we have to start thinking. We have started talking when we are seeing such a nice gene expression program is coming out. What's going on? And as Sue and Home knows, we talked to Dr. Catherine Fong last week, and she's the one who does a lot of peripheral arterial disease, limb amputation and surgeries. And she I mean, she said that a lot of these patients actually, in fact, seems like have problem with sleeping. And maybe it's perhaps because they get severe, limb pain, etc., because of the ischemia they're going through. But it's possible that we don't know whether it's because of the disease that, after the fact that you get this, but we don't know. I mean, the cause and effect, of course, but we would love to know.
Yeah, very cool. Okay, good.
Thank you, everyone. Let me stop at this point and maybe interject. I'm being notified by Randy, who is helping to run the session, that we need to take picture. I'm not sure exactly what that means, but Randy, if you want to jump in, can everybody turn on your cameras so that I guess we are taking a picture?
Yes, thank you, everyone. I'm just going to take a screenshot so that this can be loaded up for this section. Thank you for your cooperation. And that was a very good one, Dr. Ahmed. Thank you.
Wait, one of the speakers is not on my screen, Randy, so I believe Anja.
Okay.
Zagotich is, I hope I pronounced your name correctly.
She's there. I see.
Yeah, hi, I'm here.
Okay. So I guess, Randy, you just want to make sure that all the speakers' pictures are present in your screen. I don't know how you're doing this, but.
Okay, maybe after we've done with the participants, we can now have another one that is going to have only the speakers alone.
Okay.
How about that?
Sounds good.
Yeah.
So maybe at the end of the session, if you guys can stick around for we try to make it quickly. All the speakers, we can just collectively make one big happy picture. Okay, great.
Yes.
Mohamed, I have like 10 other questions for you. Whether there's a differentiation between male, female, what exactly the speculated mechanism of the reduction of the calcification, is it the host that it is somehow the genetic expression changes reduces the calcification, or is it actually the EDTA nanoparticle somehow? But I'll ask you those later.
Those are interesting questions.
Yeah. Well, thank you very much. We're going to move to our next speaker, Dr. Sue Lesner. Dr. Sue Lesner is also another collaborator we have worked with quite a bit. Dr. Lesner is an associate professor in School of Medicine. She is also a faculty in our biomedical engineering program. And so we have collaborated on a particular project for the past 2 or 3 years. And I'm going to let her to take over from here and present her work. Sue, I'll give you a 10-minute and a 5-minute notification. That's assuming your 5-minute question answer time is preserved at the end. Thank you very much. It's all yours, Sue.
Let's see, my display is not coming on correctly here.
I see your PowerPoint presentation, but it is just not in the presentation mode.
Yeah, it's actually not responding. Let me stop sharing and put it into presentation mode first, and then try sharing. My screen seems to be frozen.
Yeah, the computers just have a sense of when is the best time not to cooperate.
Yeah, this is really bizarre because I haven't.
So if you think it would help, maybe we can move to the next speaker, come back to you.
I might need to log out temporarily.
Let us start with the next speaker, and it should be a 15-minute speaker. So if during that time you can, resolve the problem and join us again. Is that okay?
Yeah.
Okay, all right, great. So Anja, it's all you. I don't know you, so please introduce yourself. And if you can tell me how to pronounce your name, please.
Yes, absolutely. Hi, everyone. My name is Anya. I am a PhD student here at the University of South Carolina, and I'm in the Department of Epidemiology and Biostatistics, doing my PhD in biostatistics. And today, let me see if I can set up my screen share. Today, I will be sharing some research and some results, as well as data analysis from some dissertation work. And let me see here if I can make this full screen. Are you able to see my slide big on your screen?
Yes, we see it fine. And I will notify you 5 minutes prior your talk ends. So at 5 minutes, and then I'll let you go till about 12 minutes, give you 3 minutes of questions, answers.
Wonderful. Thank you. Thank you so much. All right. Well, thanks everyone for being here. I'm really happy to be presenting. This work is joint work with my advisor here at the university, Dr. Alexander McLean. I'll be telling you about a novel approach to high-dimensional linear regression that we've been working on, as well as how this can be used in the pharmacogenomic setting. All right, we'll start with a background on high-dimensional linear regression. We'll go over the approach as well as an example with drug response data, and then any questions you might have. So I think most of you will be familiar with linear regression, where we have a matrix of data. Here we call it Z, and some coefficients and the regression coefficients. And the particularity of high-dimensional data is that M, the number of predictors in the model, is much, much larger than N, the number of observations. And there have been many methods developed previously to select what variables are important in this high-dimensional setting. And lasso is a very popular one, so is ridge, and so on and so forth. And that's in the frequentist perspective. In the Bayesian perspective, the typical approach for high-dimensional regression is to put a prior distribution on the model space, meaning on whether these beta coefficients are going to be null or non-null. And that's how variable selection is being done. One challenge with these two approaches to variable selection in high-dimensional settings is the challenge centers around inference. These methods are able to select the variables, but do not necessarily estimate variances, and therefore do not provide test statistics and p-values, making the inference difficult. Other post-selection tests have been proposed. So there are separate things we can do after the initial modelings. The initial modeling's been done. And when grouped together, sometimes people call this double selection approaches, where we're trying to simultaneously do the model selection and inference. And so in this new framework that we'll be developing, I'll be talking about doing this simultaneously. Another thing that's often being done in the predictor much larger than observation setting is to fit many univariate linear models. And by doing that, you're solving the inference piece. However, you need to be adjusting for a score of some kind of the remaining predictors, and that brings its own challenges. You need really strong assumptions to hold to avoid bias. And this is also computationally demanding. And so to address these challenges, we've developed, still in the univariate perspective, we've developed a way to treat the effect remaining predictors as a latent confounding variable, and still focusing on the univariate perspective, so focusing on one variable at a time. And so when identifying a remaining confounding variable, we treat it as missing, and as a result, we're able to use the EM algorithm to estimate this latent confounder, and we condition on this to get predictions. And so we call this approach unhide-them, univariate approach to high-dimensional linear regression via the EM. And so this is a predictive approach. It gives us predictions, just like the penalized regression models that we saw earlier, the variable selection models. However, one nice thing is that we've also developed the inference within the same framework. And in terms of this inference, what people have often done is inference on the coefficients themselves, but there's also the predictive inference. So when we, once we have estimates of regression parameters and we get predicted values for some new data set, Z new, we are able to formulate confidence intervals around the expected new predicted value and also prediction intervals. So one way that has been done before is using distribution-free methods, particularly conformal inference. And that is kind of part of this post-selection tests that I've been mentioning. It refits the penalized model multiple times. It's distribution-free, so people identify a desired coverage level for their confidence or prediction intervals, and then find bounds, upper and lower bounds, to get, for example, a 95% prediction interval or confidence interval. While conformal inference has some really nice properties, finite sample coverage properties, it can be really long to run due to refitting. And so in unhide-them, we're combining the sparse linear regression, the variable selection part, with confidence and prediction intervals for predicted values. And so what we do more specifically is, in the traditional linear regression, we split the equation by focusing on one predictor ZM at a time, and we collapse all the other predictors into ZM, a missing latent confounder. And what we're able to do with the E, we're treating that as missing. We're using the EM algorithm. This is the typical formulation. We're taking the expectations of the log likelihood conditional on parameters that have been maximized, estimated via the MLE at a previous iteration. And because we're working with an exponential family and we're using the EM algorithm, we're able to rely on first and second moments, which are the sufficient statistics of this missing variable. And using some assumptions, we're able to very easily calculate these quantities. And so that helps us with the computational burden. And that's for the E step, that we're leveraging these nice properties of the EM. In the M step, we've also been able to formulate something that looks fairly simple to get estimates for coefficients for the model. We're able to do something that looks like least square regression, where, however, once you break it down, you see that we have our predictor of interest and the latent confounder. And also in this step, we're able to get covariance matrices. So we're calculating variances to do inference later on. Something that we also do in the algorithm is to help convergence. We have an additional mixing step after the M step that helps us converge appropriately at an appropriate speed to the estimates. And so the final element of the algorithm is the prediction part. So once we've estimated W, the latent confounder, we finally relate that to the outcome Y, and this is where we get predictions. And in this final model, we're able to get variance of coefficients and residual variances. And using those, we're able to get something really important, which is the variance around a new prediction. And using that variance, we can write confidence intervals for a prediction. And using that variance plus the model residual variance, we're able to write prediction intervals for a new value. And so now I'm going to show you how we've used this in an analysis of an anti-cancer drug response in cancer cell lines. You've all heard the terms precision medicine, or previously known as personalized medicine. Big data and high dimensional data have been tools that have helped us develop precision medicine a lot. And these high dimensional models that I've mentioned, whether on HiThumb or other variable selection model, can really help us pick out what genomic traits of a person impact outcomes, but few of the traditional high dimensional models have this predictive inference capability. And in pharmacogenomic scenarios, this predictive inference can really guide treatment choice. And so in this analysis, I'm going to show you how with HiThumb, we've been able to generate accurate predictions and prediction intervals of drug effectiveness for patients based on drug genomics. Based on genomics, as opposed to just, for example, clinical data. So the data set comes from the Cancer Cell Line Encyclopedia, which had 8 drug data sets, so 8 different drugs. For each of these drugs, we have a drug response outcome. And for each of these drugs, the observations are 491 cell lines of a human, and the predictors are nearly 20,000 human gene expression levels. Drug response, more specifically, is the area above the dose response curve, as defined traditionally in such studies. And so we're comparing on HiThumb for linear regression with the traditional variable selection methods. Everywhere we've used tenfold cross-validation. If we needed to tune parameters additionally, we did another tenfold cross-validation. For predictive accuracy, we're comparing mean squared predictive error. And for prediction intervals, we are comparing coverage probabilities. For the MSEs, we're comparing it to lasso and the other variable selection methods. For the prediction intervals, we're comparing to conformal inference. And so very quickly here, when we look at prediction error, for 4 out of the 8 models on HiThumb performed really well, dominating about half of the models, with SCAD and lasso in second position. So this is saying we've got on par predictions with current methods. In terms of the prediction intervals, for the coverage probabilities, they're all similar, very close to 95%. That's the desired output for all of these. However, something that we see is that for on HiThumb, the width of the prediction intervals is very similar across the cross-validation folds. And so this is desirable because the cross-validation folds are chosen at random, and so we want it shouldn't matter what cross-validation fold you are, it shouldn't matter your prediction interval length. And so what we see from these results is that we're able to predict well, but we provide accurate and more consistent prediction intervals. And in this pharmacogenomic setting, this is high value information to patients and providers, and it helps make decisions. For example, if you have 2 drugs that have a similar predicted effectiveness with similar prediction intervals, providers can suggest to pick one or the other based on other clinical factors. Or alternatively, if you have 2 drugs where the effectiveness can be a bit different, you can see how much the prediction intervals overlap, and then again, that informs your decision. And so in general, more from the methodological side, current methods provide all of these elements, but separately, and you got to mix and match. But here we've been able to build a bridge to gap between the methods that do variable selection and inference separately. And today I've presented from a frequentist perspective. We are also developing this in a Bayesian framework. And later on, if you see me throughout the conference and you're interested in learning more, I have the other nice data analyses with interesting results that I'll be happy to share with you. Some references, and I look forward to hearing your questions later on. Thank you so much.
Thank you, Anya. That was very nicely done. I have actually several questions for you too, but I don't think we have time.
Okay.
I'm sorry, everyone. I think we're going to have an opportunity to interact maybe later today and also tomorrow, but please look her up if you have questions. But we do have to go, as I explained at the beginning of the session, we have to give our Zoom session over at 3:15, so we just don't have time. Thank you. Sue, did you want to give it another try? We see it.
Okay.
Okay, great. Sue, it's all yours.
Okay. All right. So I'd like to thank Dr. Valafar for inviting me to speak here. And this work is a collaboration with Dr. Valafar and Dr. Agostinelli, who spoke earlier. And we're interested in applying big data at multiple scales to the clinical problem of peripheral arterial disease. So, so do you see my second slide?
It's still at the title slide.
Okay. All right, there's a problem with this. I think this is the same problem that Forrest had.
Yeah, I wish we had him here so he could tell us how he fixed it.
Yeah. Let me try something else. Yeah, there seems to be an issue with the using PowerPoint with this. We'll try this.
This is how Mohamed Azar was working. So if this is going to work for you, this will be fine.
So did it switch to the second slide?
Yes.
Okay, great. So just to introduce the problem of peripheral arterial disease, this results from atherosclerosis in the arteries of the lower extremities. And the problem is really that when you have an atherosclerotic lesion, it can partially block off these large arteries, resulting in ischemia in the downstream tissue or insufficient oxygen supply. And in the less severe forms of the disease, the presentation clinically is what's termed intermittent claudication, which is pain in the extremities during exercise. In the more severe form, which is termed chronic limb-threatening ischemia, there is pain even when the patient is not exercising, and ultimately the reduced blood flow can lead to non-healing ulcers or even gangrene, resulting in a possible need for amputation. And so there are several approaches to grading this condition clinically. These are two of the traditional scales, the Fontaine and the Rutherford. But you'll note that in the earliest stages of the disease, this condition is asymptomatic. And then as it progresses, you get different degrees of claudication, eventually giving rise to rest pain, which is pain without exercise, and then potentially progressing to a tissue loss and gangrene. And there are current approaches to developing new methods to classify peripheral arterial disease based on predicting the patient outcomes after treatment. So these older approaches are not that useful to the clinician to determine how best to treat the condition based on combinations of the patient ischemia, tissue damage, and so there's a newer method that has been published recently called the lower extremity threatened limb classification system, developed from the Society for Vascular Surgery. So why do we care about peripheral arterial disease? Well, in the state of South Carolina, this is a huge issue. In fact, our state has among the highest rates of non-traumatic lower limb amputation of anywhere in the country. It's well above the national average. And one of the reasons for this is that we have a large population of people who have risk factors for peripheral arterial disease, including hypertension or high blood pressure, hypercholesterolemia, which is high blood cholesterol, diabetes, smoking, and chronic kidney disease. So all of these conditions are elevated in the state of South Carolina, leading in part to these very high rates of peripheral arterial disease and lower limb amputation. And additionally, there's a concern, and this was mentioned nicely in Dr. Azar's talk, is that there are few non-surgical options to treat peripheral arterial disease. So mostly the condition is silent up until the patient develops intermittent claudication. But at that point in time, the disease is already well enough advanced that there are relatively few medical approaches. Now, if the patient is diagnosed early enough, the main approach to limiting the extent of the disease is to control these modifiable risk factors. So these are the same risk factors that are typical for coronary artery disease. And many of these are controllable, such as high blood pressure, high blood cholesterol. Patients can be counseled to stop smoking. But for the most part, oftentimes by the time the disease is diagnosed, it's already quite advanced, and the only approaches to treating it are surgical intervention, either endovascular intervention such as balloon angioplasty, or open surgery such as femoral endarterectomy. And also thank Dr. Azar for introducing everyone to the notion of vascular calcification. So this is a further complication frequently seen in peripheral arterial disease, especially in patients who have chronic kidney disease. And in this, in a calcified vessel, the vessel becomes almost bone-like. So the example in the upper right is a endarterectomy specimen taken from a patient undergoing femoral endarterectomy. And these excised plaques can be as hard as bone. And calcification, as Dr. Azar mentioned, can occur either in the inner layer of the artery or in the middle layer. The mechanisms are thought to be different. But more importantly, from the perspective of the surgeon, it complicates the endovascular treatment. So one approach to endovascular treatment is angioplasty, where a balloon-tipped catheter is inserted into the vessel, and the balloon is expanded to try to open up the vessel and restore blood flow. This doesn't work if the vessel has become heavily calcified. And it also complicates open surgery as well, because the surgeon has to deal with a vessel that has basically turned into a tube of bone. So the image on the lower right is a patient CT scan. The brighter areas indicate calcification. So these are in fact the bones of the pelvis. But here are a couple of the femoral arteries, and you can see these white regions indicating calcification in those vessels. So it's been known for quite a few years that coronary artery calcification is actually a very useful tool to predict outcomes in patients with coronary artery disease. So coronary artery calcification is measured using something called the Agatston score. And this Agatston score predicts not only severe outcomes, cardiovascular outcomes, such as myocardial infarction, but it also predicts all-cause mortality. More recently, there's been evidence published in the literature that indicates that calcification in the lower extremities is also a useful predictive tool, not only for adverse events in peripheral arterial disease, but also for all-cause mortality. So we define adverse events in peripheral arterial disease using the acronym MAIL, which stands for major adverse limb events, and that includes a death, amputation, major amputation, or the need for reintervention following the initial surgery or endovascular treatment. And these recent studies indicate that lower extremity calcification can serve as a prognostic tool for these major adverse limb events. So after we became interested in looking at this issue of could we develop a similar score for calcification in the lower extremities, and would this actually be useful to predict adverse events in patients with peripheral arterial disease? And this led to, we started an IRB protocol in 2018. Our current collaborator at Prisma Health is Dr. Catherine Fung, and this work is being done with Dr. Valafar and Dr. Agostinelli. We have 29 patients enrolled to date, and we've had a conference paper published trying to use machine learning approaches for automated analysis of femoral artery calcification as a potential prognostic tool in peripheral arterial disease. So the aims of this study were first to develop and train an automated calcification detection system for the femoral popliteal circulation using current machine learning techniques and analysis of patient-specific CT images. So these CT images are typically acquired during diagnosis of peripheral arterial disease patients. So we were using only data that is already available in treatment of these patients. Secondly, we wanted to determine if there's a correlation between femoral artery calcification and a panel of circulating biomarkers. And thirdly, we wanted to determine whether vascular calcification, possibly in combination with these plasma protein biomarkers, could be used to predict the outcomes in patients undergoing femoral endarterectomy. So the timeline for the study is shown here. So patients are referred to the vascular surgery clinic at Prisma Health Midlands. They've if they have not already undergone a CT scanning, they will undergo it there, and then they're consented into the study. At the time of their CT scan, we get the CT scan at over here at USC, and then at the time of their femoral endarterectomy surgery, we get the excised plaque specimen as well as a blood sample. And the blood sample is used for a biomarker analysis. The plaque sample is used for a micro CT and calcium analysis. Importantly, these patients are followed after the surgery, and we obtain their clinical records and also some quantitative information, particularly the ankle brachial index. And that's just a measurement of the blood pressure, the ratio of the blood pressure at the ankle to that in the arm. This is indicative of lower limb ischemia. So anything below a level of 0.4 is considered chronic limb-threatening ischemia, but anything less than 1 indicates some extent of ischemia. So we get this information from the patients on multiple follow-ups, so we can try to relate the calcification score and the protein biomarkers to patient outcomes in this study. So essentially, we're looking at a problem where we have a wide variety of data for each of these patients, including information from their electronic medical records, plasma protein biomarkers, a calcium score from their CT scan, and then we want to use all of this information to try to predict patient outcomes in terms of whether they have a beneficial outcome and their limb function is restored, or if they have an unsuccessful outcome, which I've already defined as male major adverse limb events. So the first part of this work is essentially looking at the macro scale or tissue level scale by performing image-based calcium scoring. And to establish our ground truth, we take the individual patient plaques and we do micro CT to develop a volumetric calcium score. Then we extract calcium from the plaque. And you can see that the volumetric calcium score is a very good, there's a very good correlation with the actual calcium content of the plaque. Now, when we compare the same analysis for the clinical CT, we also see that there's a correlation between the volumetric score and calcium content, but the sensitivity is less. And this is because clinical CTs don't have as high resolution as micro CT, but of course, clinical CT is what is available when you don't actually have an excised plaque. Then our initial approach to calcification scoring was, all right, let me see if this movie will work. Well, I may not have my CT movie here. It doesn't seem to be playing. All right.
So Sue, try turn off the laser pointer. That might work.
What? Turn off the laser pointer?
Yes.
Oh, okay, turn it off.
Okay, now go try click.
No, still didn't seem to.
Hmm.
Will it work if you get it out of the presentation mode?
Well, we'll
If it does, give it a try if you think. If not, Maybe you can just verbally describe it and move on. We have only about 7 minutes left if we skip.
Yeah, okay. So, so unfortunately, my CT movie is not working. But what we did initially was just to use image segmentation to identify the blood vessels. So these patients, when they undergo CTA, they're injected with a contrast agent, which allows you to visualize the blood vessels. And then the calcified areas appear as higher intensity regions. And the initial approach was just to use an object tracking algorithm to follow the blood vessels through each slice of the CT, and then to segment out the calcified higher intensity areas. So there are some disadvantages to this approach, or challenges. The first disadvantage with any type of image-based analysis like this based on clinical images is that to get our validated or annotated data is tedious and requires knowledge of anatomy. And secondly, we need to be able to segregate or separate the calcification from other high intensity structures such as bones. Finally, in patients who have severely blocked arteries, the contrast agent doesn't necessarily reach all parts of the vasculature, so there may be loss of continuity in the object tracking approach. And then there are also the issue of artifacts such as previous surgical implants, whether stents, orthopedic screws, etc. We're now taking using different approaches such as convolutional neural nets to try to segment the calcified areas. But I'll move on and say something about the other side of the study, which is looking at big data at the molecular scale by analyzing plasma biomarkers. So initially we did a screening of patient samples using a unbiased large-scale antibody array of 1,000 proteins. This allowed us to identify some candidate biomarkers by comparing patient plasma to that of non-peripheral arterial disease controls. So we identified a number of candidate markers, such as osteocalcin, which is elevated in patients versus controls. This protein is known to promote energy availability in myocytes. So patients with peripheral arterial disease have skeletal muscle myopathy in the extremities because of this long-term ischemia. And then some other markers such as fetuin A were significantly reduced. So fetuin A is known to be a calcification inhibitor, and that's significantly lower in peripheral arterial disease patients, which, as you might expect, they have a significant calcification compared to normal individuals. So some of the challenges we face in this program are how to synthesize this multimodal data, including biomarker levels, clinical data from electronic medical records, and calcium scores, and how to go about normalizing data when some of it is continuous and some of it is categorical. And secondly, how do we best define the outcomes? As I've already mentioned, we can use a qualitative measurement such as the major adverse limb events, or we could use quantitative approaches such as looking at a percentage change in the ankle brachial index between clinical visits. And this is complicated by the fact that there are multiple clinical standards in use, and we need to basically work with our clinical partners to try to translate the visit notes from the electronic medical records into these different categories that are used to define the progression of the disease. So recently, as I mentioned, we're taking several approaches to improve the measurement of calcification. Doctors Valafar and Agostinelli are beginning to create synthetic data to overcome the limited supply of annotated CTAs for training machine learning algorithms. And we're using more advanced machine learning models such as LSTM and UNet, and in part using the results from the object tracking studies to help train these machine learning models. In terms of the biomarker analysis, we're continuing to identify and categorize patient outcomes and doing some dimensionality reduction and clustering analysis using several approaches such as k-means and UMAP. And I'll just show a couple of examples from that. This is a UMAP analysis of using 6 of the plasma biomarkers, age and sex as variables. And we see a clear separation for our control patients, but the peripheral arterial disease patients are kind of all over the place. These 2 in the lower left are 2 that we knew had adverse outcomes at the time of this, at the time that I did this analysis. Another approach that we've taken with this data is to do UMAP with clustering by density-based merging. This doesn't completely segregate the patients by outcomes, so we do get a good separation of the controls, which are shown here. And these are the patients with major adverse limb events, so they're not completely separated by cluster from patients who did not have adverse limb events. Then using a k-means clustering, my student showed that we could separate most of the patients with adverse events, and we saw significant differences in some of the biomarkers in these 2 clusters, but only in this example, 89% of patients were correctly grouped by their outcomes. So we still have a lot of work to do on dimensionality reduction and clustering to try to effectively categorize patients by outcomes. And so in the interest of time, I'll sum it up there. Just to bring to your attention that this peripheral arterial disease is a major clinical issue in South Carolina, and better prognostic tools are needed to risk stratify patients prior to their vascular intervention. So the message that I get from the surgeons I work with is that they can generally achieve success in terms of the surgery, but the problem is they don't know which of those patients will remain stable long-term and which will progress and go on to require either additional intervention or an amputation. So we need better predictive tools. We think that machine learning approaches are going to allow us to automate calcium scoring in the lower extremities to provide a simple tool for clinicians to use, similar to the Agatston score in the coronary circulation, and that the dimensionality reduction and clustering of this multimodal data set shows promise as an approach to classify peripheral arterial disease patients by their longer term outcomes. So with that, I'd be happy to answer any questions. Thank you.
Thank you, Sue. Thank you very much. I'm sorry for the technical difficulties, but it happens these days. That's part of the nature of the game. But we do My apologies, but we do have to get going because this session officially ends at 3:15 and we have to hand over Zoom to somebody else.
All right.
So I'm not sure what to do with the picture taking session, but Aaron, you're up. Let me give a very brief, quick introduction. Aaron Hines is a PhD student in the College of Engineering and Computing, Department of Computer Science and Engineering. He's working in the area of protein structure, and I'll let him introduce his work. Aaron, I'm going to let you know 5 minutes ahead of time before you have time. I'm going to assume there's no question answers. Please feel free to contact any of our speakers to discuss any of your questions. I encourage you, of course. But Aaron, it's all yours.
Thanks, Dr. Valfar. So my name is Aaron Hine, as Dr. Valfar had mentioned, and today I'd like to talk a little bit about protein data mining optimizations in PDB-Mine. So kind of the motivation for this work is we're all familiar with the importance of protein structure and protein structure calculation, and even though there are multiple ways to go about this, often it involves predicting the dihedral values for each residue or amino acid residue in the chain. So this is something that's been studied for a number of years, and even back in the 1960s, Ramachandran came up with these plots that help us visualize possible dihedral values for various amino acids. And what we're trying to do in this work, or with PDB-Mine, is to basically go query the worldwide protein data bank that has hundreds of thousands of proteins in there, so that we can find matches to further restrict this space and get a better idea of what these dihedral angles are. So PDB-Mine is hosted, it's an online tool hosted on festus.cse.sc.edu. And what it is it's a tool that allows us to query for various sequences of amino acids throughout the protein data bank. So PDB-Mine, when you put in a sequence, you give it a window size. And what the tool does is it takes that sequence and it splits it into windows of the window size. So let's say that you had a sequence of 100 amino acids. It would split those 100 amino acids, and it would take the first 7 starting at position 1 to 8, and then 2 to 9, and then 3 to 10, and so on and so forth. And for each of those windows, it then queries the entire protein data bank for any matches, right? So this query that I have typed in here is for 1 RX2, and then a window size of 7. So it would take the first 7, second 7, so on and so forth. So a couple caveats here when you're choosing the window size. Smaller windows, if you had a window size of 1, that's basically the Ramachandran plot. It's going to find every possible value in the protein data bank for just that one amino acid. Larger windows can be so specific that they don't return any results because there isn't that exact sequence that can be found. So a little bit about PDB-Mine and how the performance of it is. Each frame, each in this case 7 or string of 7, sequence of 7 amino acids, take a constant just under 1 second to run. And so the larger the query sequence is, the more it takes, but it scales very well. And then once that query is complete, the results can be retrieved from the website either as a JSON document or as a collection of CSV files. So if you choose JSON, it looks something like this, where you can see we have the frame here, or this query was done with 5, but you can see the 5 amino acids, and you see the first match, the protein, the chain, the second match, protein, chain, so on and so forth. And then for each protein and chain, it gives a selection of residues and then the phi and psi angle for each residue. If you choose the file results, we have a file. We produce basically a folder with files for all of the proteins and what the results were, as well as a summary file with all the proteins. And then actually for the residue, we combine everything. So if you had a window size of 7, there would be the first 6 plus the residue you were looking at, the first 5, the residue you're looking at, and the one after it, and so on and so forth. So those are all combined here. Apologize for going so quickly just because of time here. So you can see here the query that I showed earlier. When you run with a window size of 5, you can see approximately how many hits are found in the Worldwide Protein Data Bank for each frame. So you see some have as many as 800 hits here, others have very few hits. And that changes obviously window by window. Like here's the window size of 7. You see that there are much fewer matches. The most is, instead of 800 matches, here you see 250 is the most number of matches, and many less than that. And then, just to kind of wrap up here, remember this was the Ramachandran plot of glycine. So because of there's not really a side chain there are phi and psi possibilities across all 4 quadrants. So it's a very broad space. And so that the query that I showed earlier, if you see the Ramachandran plots or the plots generated from that query, and the red is the actual value from the known protein. But you see with a window size of 5 here, that search space is dramatically reduced. And with a window size of 7, you can see almost all of the possibilities are reduced even further, but there's a very strong collection around what the actual value was. If you're looking at the glycine at position 66, which has different amino acids on each side of it, you see again a lot more. Here there's not as big of a reduction, and there's a lot broader scattering, but you can still see the strongest the strongest concentration of values here by what the actual value is. Again, so I'm down to 2 minutes. I apologize for going so quickly here, but the challenges here were to reduce the query size or reduce the query time and get a good result set. Here are some references. I know we only have a minute left, but I would love to get questions. Feel free to contact me after the presentation, or anytime after the conference or during the conference. I'd love to talk more about it. So, thank you.
Thank you, Aaron. Actually, to my calculations, we still have 5 minutes left, but I appreciate leaving some time open here. Maybe we can complete the picture taking. And if there are questions, we should be able to entertain one question for any of our speakers. But let's get the picture done first, so that way we have all the necessary items out of the way. So, Randy, how do you want to do this?
Have all the participants with the speakers, and then I'll now take the speakers alone.
Okay. Can everybody who wants to participate in the picture, please turn on your camera? That includes me.
Okay, so let us all put on a smile with a beautiful hair. Okay, so here I go. Then I'll take one more. Thank you. So can I have the speakers alone?
Am I a speaker? Should I turn off my camera? I'm not sure.
Okay, Dr. Alpha, can you brighten your, it's a bit dull on your side.
Wait, you mean my background?
Yes.
Not my personality, right?
Uh-uh.
Okay, the problem is that I have, if I remove,
okay, I think it's much better. Yes. Thank you. So I'll take one more. Okay. Thank you all.
Well, thank you, everyone. I have to turn this over. I want to thank again in the last minute all the participants and all the speakers. I very much enjoyed hearing the latest, and I am certainly going to follow up with you guys to see if I can answer your questions. Please feel free to reach out to any of the participants and the speakers to discuss any of the questions further. Thank you again. Bye, everyone.